Here are some questions from microbiology and pathological aspects regarding hepatitis virus infections, and we will be discussing regarding that. Firstly, we know that hepatitis virus are of 6 types: hepatitis A, B, C, D, E, and G. F is not present, and each of them has their own families, their own serological diagnostic markers, and their genetic materials and such. We know that hepatitis A and E virus are transmitted from fecal-oral route, and that they generally cause acute hepatitis. And we also know that hepatitis B, C, D generally cause chronic hepatitis, and that they are transmitted through vertical transmission from mother to fetus, from blood transfusions, from drug abuses, from unprotected sexual intercourses, and also sharing of personal items and hygiene matters. Besides this, we also know that HA virus and hepatitis E virus are in non-encapsulated, whereas hepatitis B, C, D are encapsulated. And one of the most key features is that all of these are all of these contain single-stranded RNA genetic material, whereas hepatitis B virus contains partially segmented, partial double-stranded DNA material, which is what makes HPV so different. And all of them each have their own serological diagnostic markers. Let us begin with the hepatitis B virus. Hepatitis B virus presents with surface antigen, core antigen, but we cannot see this core antigen in the laboratory diagnosis because it is hidden. Except this, we have pre-core antigens, and we also have respective antibodies to the antigens, which have surface antibodies, pre-core antibodies, and for core antigen we have two antibodies: IgG antibodies, which are present at a later stage, and IgM antibodies, which presents at the earlier stage. Besides this, we can also see HPV DNA polymerase and HPV DNA through molecular methods. Plus, we can also do liver function tests regarding bilirubin, aminotransferases to see the hepatic function. We also have one extra information regarding incubation period. Since we know that A and E are acute infecting, so its incubation period is quite short. We can say from 2 weeks to 6 weeks, or 4 weeks to 6 weeks. And for HBV and HB virus, hepatitis B virus, C virus, and D virus, it's almost similar, from 2 weeks to 26 weeks, or from 4 weeks to 26 weeks, which is almost 8 months to 9 months for incubation period. We will see later why this has such a chronic stage. Now we will go towards the morphology of hepatitis virus, and this is a quite simple schematic diagram for the morphology. We can see outer circle, purple circles, those are the surface antigens, and the inner red ones are your core antigens, and between this the green plus signs, they are your envelope antigens or pre-core antigens. Inside there is a hexagonal capsid, and inside that we have double-stranded partial, see, it's not complete, it's partial DNA material, and there is a DNA polymerase. Now if we go through the electron microscopy, this is the picture that will be seen. You can see some spheres here. Those are your hepatitis B are normally found in 3 morphologies: sphere, filamentous, and Dane particles. So right here, if you see the circles are your spherical particles, the filaments are your filamentous tubular particles, and these circles are your Dane particles. Now we will go further into what these look like. You can see on the net that there is different kind of variations. Now this is ideally the schematic diagram we presented earlier is the Dane particle, this is the filamentous particle, and this is the spherical particle. That was it for the morphology of the hepatitis virus. Also, I forgot to mention regarding the serological diagnosis of other viruses except the HPV viruses. And it's pretty easy because you can just write the RNA material of that particular virus, and since not all of them presents with surface antigens, you can just write, suppose for HIV virus you can write HIV virus antigen, this virus antigen, and for antibody HIV virus IgG antibody, IgM antibody. Now for A and E, normally you do not write the G antibodies because these are the acute infectious viruses, but for other viruses you can write G and M antibodies, since all of the chronic viruses can cause acute infection and then go through resolution or go progress to chronic, but A and E virus cannot cause chronic viruses. That's to be remembered. And okay, now we will move forward to the actual concepts that I wanted to talk about in this videos. So why does this hepatitis virus only infect the hepatocytes? So the reason is that hepatocytes present a receptor which is called anti-CP receptor. Full form is sodium taurocholate co-transport polypeptide receptor, which is a bile salt sequestrants or whatever. It also lets the hepatitis virus inside the hepatocyte. Now once the hepatitis virus is inside the hepatocyte, then it, through viral polymerase, the DNA polymerases, it forms into a circular DNA. It was partially double stranded DNA. Now it converts into a conventional circular DNA. And now this DNA undergoes to transcription. Now after transcription, it forms two kinds of materials. One is mRNAs and another is pre-genomic RNAs. Now this pre-genomic RNAs, through after reverse transcriptase, they form the genomic DNA materials, and the mRNAs are then translated into proteins of the genetic material. Now we have the DNA material and we have the proteins. Now they both are combined into a virion, and the virus replication occurs like this. So this is how the viral replications occur inside the hepatocytes. Now how does it cause inflammation, infection, hepatitis, inflammation? So the MHC1 receptor of the hepatocyte presents this antigens on its receptor, and the macrophages, CD4 plus cells, CD8 plus cells sense this, takes these antigens and attacks to the liver hepatocytes. Now so the CD8 plus cells, CD4 plus cells are actually the one attacking the liver and causing the inflammation and necrosis there, not the hepatitis virus, though it is the flame lighter or whatever. Another question I had was, why does HAV and HEV only causes acute hepatitis, and why does the other ones cause chronic hepatitis? Now the reason is that the infectivity of the virus, or you could say the virility, pathogenicity of the virus. So this might not be entirely correct, but I will tell you from my logic. So what happens is that when the hepatitis A or E infects your body, then your immune cells recognize it. The antigenic particles are very virulent. They have high virulence capacity. So our immune cells recognize it and attack to it immediately, and then cause the resolution of the infection. So our body is able to fight against the virulence particles from the hepatitis A and E very good amount, right? But for the chronic infections, what happens is that the actually from besides what you think, let's take HPV for example, and you can say the rest are same. So what happens with HPV virus is that we know that it secretes tons of hepatitis B surface antigens, but these surface antigens are virulent particles, so like they are not that infective, you might say. So our B cells, they do produce the antibodies against it, but whatever the antibodies are produced against it, then they form complex with the antigen and they are undetectable. So there are lots of antigens and our immune cells kind of develop a tolerance for it. They are like, I can't deal with this anymore, and just leave them alone. So antigens are present everywhere, and that's why the antibodies are not made against it, because the immune cells are done with them. They don't want to deal with them. They have developed tolerance. But when after the infections has resoluted, they do still attack the hepatocytes and stuff, slowly, and then when they are resoluted, when the antigens have been all destroyed and ingested, then only the B cells, the immune cells, regain their motivation, and then they start developing the antibodies, which is the reason why the antibodies develop only after the infection has resided or the antigens have been reduced. Okay, and I did have this question, like normally the antibodies should have been made against them within 2 weeks, and they should have attacked the antigens and neutralization should have been occur. Why isn't it occurring here? But as I mentioned earlier, it is occurring, but it is occurring in very low amount, so it is undetectable, and high amount is not have been occurring. Next question is which is more dangerous, acute or chronic hepatitis, and how does this chronic hepatitis set the stage for hepatocellular carcinoma and cirrhosis? Now, I would say that acute hepatitis is actually more infective, more dangerous than chronic hepatitis. Number one, because acute hepatitis is causing एकदम धेरै immune responses. It's causing massive immune responses. So massive hepatocyte necrosis is occurring, necrosis, inflammation is occurring, and only few cases are severe enough to cause fulminant hepatitis. So the immune response is actually more in acute hepatitis, so it appears as if it's more important and more dangerous, but it's actually the chronic hepatitis which is more important, because whereas it might not develop as much as clinical symptoms, dangerous effects as the acute hepatitis, but in the long term, since virus DNA keeps using Thore human DNA and replicate, it causes defects in the genes of the hepatocytes, the normal nuclear gene. It also disrupts the pathways. It causes some suppressions in tumor suppressor genes, TP53, and some other mutations, I think microsatellite mutations and whatnot. And these mutations accumulate, and then they cause the hepatocellular carcinoma, and hepatocellular carcinoma, if for a long period of time, then they cause cirrhosis. So this is how it causes the so in the long term, chronic hepatitis B is more dangerous. But if you ask me on for a short period of time, like let's say a person has been having 6 months of acute hepatitis and 6 months of chronic hepatitis, then obviously the acute will have killed you by then. It's okay. Next is we will move on to the pathology. What's the difference between the acute and chronic hepatitis morphology? The rest are quite similar. The only difference is that in acute hepatitis there is massive necrosis, massive inflammation is present. So obviously what will you see in under microscopy? Plasmacytic infiltration, lymphocytic infiltration, you'll see necrosis. And we know in hepatocyte there are quite a few types of necrosis. You can mention everything there: bridging necrosis from portal triad to portal triad to central venous triad, centrilobar pericentral necrosis, and then periportal necrosis, interface necrosis, lobular necrosis, whatever necrosis that comes to your mind, you can say. So inflammation is seen, plasmacytic, lymphocytic infiltration, and apoptosis can be seen, apoptotic acidophilic cells can be seen. You can also write ballooning degenerations can be seen. Overall, this is the basic. And for chronic hepatitis, so we know that the necrosis is occurring in chronic. What happens in chronic? Fibrosis occurs. So the thing you should not miss here is fibrosis. So moderate to marked inflammation is also seen here, mononuclear infiltrates, plasmacytic, lymphocytic infiltrates, periportal fibrosis can be seen, necrosis can also be seen, and in chronic stages some part of cirrhosis can be seen. I think that is it for the microscopy. If we talk about the clinico-pathologic syndrome is quite easy, as what is number one? You have acute infection, asymptomatic phase, acute infection, symptomatic phase, then you have acute liver failure, then after that you have chronic infection, then after that you have a carrier phase. Now let us talk a bit about the carrier phase or the window period. Now remember, I had talked about how the antibodies for the surface antigens occur right after the infection has resided and HBsAg level has decreased a lot. Now sometimes what happens is, even after residual of the HBsAg antigens, the antibodies do not appear, but they do still be transmitting the infection from to other persons. Now this is called the window period. Carrier period is the same as in, like, you will see some antibodies, you will see some antigens presented. Normally you will see HBC antibodies and HBsAg antigens, and they are carrying it. Actually infection has not occurred on them yet, it might have, and they are still transferring it to others. So that's the carrier phase. Now if you see, now if you tell me what are the differences between clinico-pathologic syndromes, what is the difference between all of this, let us talk about first symptomatic and asymptomatic phase. Now in both cases the serum amino transferase levels have been elevated, necrosis occurs, and your antiviral antibodies are seen. So not microscopy, there is not much different. The only difference is the clinical. So one shows symptoms, one does not show symptoms. What are the symptoms? Nausea, vomiting, jaundice. Main is the jaundice. And I forgot to mention that in the symptomatic phase there are actually four sub phases, right? And they are incubation period, we have already talked about incubation period, symptomatic pre-icteric phase, like before jaundice occurs, then icteric phase while jaundice is occurring, and convalescence where the infection resides. Another is acute liver failure. In acute liver failure you'll see massive fulminant necrosis, hepatitis, inflammation, all of in this HB, A, B, D, E, any of them can cause this acute liver failure. We also know the non-alcoholic liver failure and alcoholic liver failure, and on top of that, basically serum aminotransferases are also elevated in here. If we talk about chronic, we know that C, D, E cause the chronic infection, and it is of two types, progressive and non-progressive. What might it progress into? Hepatocellular carcinoma and cirrhosis, or it might not progress, and it's very rare cases. All right. And carrier stages, I've already talked about that. Now I want to show you a picture of a graph. This graph is something bare minimum that I want you to remember. We have here different kind of antigens and antibody, and this is the month, period of time in months. This red one here is HBe antigen, the precore antigen. See how it is elevated from second month to a certain month, and when the level of antigen has resided, decreased, stopped, then only the antibody increases. Same for the HBsAg antigen increases, then the our immune cells attack against it, then slowly they are all removed from the body, and only after that the antibody against them are performed. Now initially pre-core antigens and the core antigens do develop, but we cannot see it in the lab, so there is no the hill for the core antigen, but we can see that the core antibodies have increased the most. This is a quick flow chart that I also want you to see how after incubation period and acute hepatitis, we know that almost all of the infections cause acute hepatitis, and most of the acute hepatitis actually resolution, they actually recover their resolution. Some stays as carriers, some develop into chronic hepatitis, 5% and 5%, and 90% recover. So it's actually a good prognosis if you ask me. Then after the among the chronic hepatitis, the people who have suffered through chronic hepatitis, 70% of them develop cirrhosis, and that cirrhosis can either go to hepatocellular carcinoma and liver failure. Cirrhosis is generally a end stage liver disease, so that basically means your liver is fucked up and it's dead already. It's not dead, but end stage. And rest hepatocellular carcinoma, and 30% of the chronic hepatitis remain as carrier, and only 2% recover. So that's it. Few pointers that I wanted to mention was the surface antigen denotes that the virus onset of infection, it denotes the onset of infection, whereas the pre-core antigen, E antigen, denotes that the virus is replicating in the body. The antibody against the IgM antibody against the core antigen denotes that the active set of the infection, and IgG denotes the chronic stage of the infection. And the vaccination that is given against the virus contains the HBsAg antigen. It contains the surface antigen. So the antibody present in our body is anti-surface antigen antibody. So if a person, if we test a person and he has HBsAg, but he doesn't have other antibodies, HBc antibody, E antibody, or other antigens, then we can say that he has been vaccinated. Thank you. That was all.